Hello everyone, my name is Fan Huang, and today I'm very excited to tell you about this story. Melanoma is the deadliest form of skin cancer that is derived from melanocytes. Current treatment for melanoma patients include MAP kinase pathway targeted therapies and immune checkpoint blockades. However, these therapies are not able to achieve a durable response in all group of patients, which drives us to search for additional treatments that can potentially sensitize melanoma to therapies. In melanoma, the two major signaling pathways, the MAP kinase pathway and PI3K-AKT signaling, converge at this spot, where the eukaryotic translation initiation factor, EIF4E, is phosphorylated by kinases MNK1 and 2. In order to study the role of this EIF4E phosphorylation in melanoma, we generated this knock-in mouse model, where EIF4E cannot be phosphorylated, and the tyrosinase CRE-ER system allows us to induce melanoma. Using this model, we firstly show that, compared to the EIF4E wild-type mice, the knock-in mice have decreased tumor outgrowth and metastasis. We also show that the knock-in tumors are more differentiated compared to the wild-type tumors. They express higher level of the melanocytic lineage transcriptional factor MEDF, along with two of its downstream targets, melan-A and GP100. Both of them are highly immunogenic melanocytic antigens. Based on this group of data, we then hypothesized that phospho-4E might contribute to a process in melanoma called phenotype switching. Basically, melanoma cells can switch from a more differentiated and more proliferative state to a more dedifferentiated and more invasive state, which allows them to resist both targeted therapies and immunotherapies. Here, using the wild-type versus knock-in tumor-derived cell lines, we demonstrated that NGFR, a key regulator of melanoma phenotype switching, is under translational regulation of phospho-4E. The phospho-4E deficient knock-in melanoma cells have decreased expression of NGFR, they are more proliferative and less invasive compared to the wild-type tumor cells. It has been shown that phenotype switching could lead to increased pro-inflammatory cytokine production. So we did a cytokine array, and we were able to identify a subset of cytokines and chemokines that are downregulated in the knock-in tumors. Among these secreted factors, we further identified CCL5 as another translational target of phospho-4E. In line with our data showing that the knock-in tumors have preserved melanocytic antigens melan-A and GP100 and decreased pro-inflammatory cytokines, we showed that the knock-in tumors have higher infiltration of T cells and lower infiltration of MDSCs, the myeloid-derived suppressor cells, meaning that they are more immune hot compared to the wild-type tumors. Next, we investigated the tumor cell extrinsic role of phospho-4E. When we injected the same tumor cells into wild-type versus knock-in hosts, we showed that after subcutaneous injection, tumors grow slower in the phospho-4E deficient background. And after tail vein injection, the tumor cells also form less metastasis in the hosts lacking phospho-4E. This group of data indicated a pro-tumorigenic role of melanoma cell extrinsic phospho-4E. We found that PD-L1 expression is significantly decreased in both dendritic cells and MDSCs from the knock-in tumor-bearing mice. And functionally, phospho-4E deficiency enhanced the dendritic cell-mediated T cell activation and also impaired the MDSC-mediated T cell suppression. So far, we have shown that phospho-4E in melanoma cells promotes their phenotype switching, with decreased expressions of melanocytic antigens, decreased infiltration of T cells, and increased infiltration of MDSCs. In addition, phospho-4E expression in dendritic cells and MDSCs regulates their PD-L1 expression, which makes the tumor microenvironment more immunosuppressive. All these features are associated with poor responses to immunotherapies, such as PD-1 blockade. Therefore, we hypothesized that systemically blocking phospho-4E with MNK inhibitors could improve the efficiency of immunotherapy. So we combined the MNK inhibitor SEL-201 with anti-PD-1 antibody. As expected, SEL-201 sensitized melanoma to anti-PD-1 immunotherapy in 3 different melanoma models, including a triple wild-type B16 PD-L1 model. And the combo group showed significantly decreased tumor outgrowth and improved survival. In conclusion, we showed that the tumor cell intrinsic phospho-4E promotes melanoma phenotype switching with decreased melanocytic antigens and increased pro-inflammatory cytokines, which blocks T cells and attracts MDSCs. In the tumor microenvironment, the MNK EIF4E axis regulates PD-L1 expression on dendritic cells and MDSCs. Blocking phospho-4E by MNK inhibition sensitizes melanoma to anti-PD-1 immunotherapy. And finally, we hope this study could help to inform the clinical development of MNK inhibitors in combination with immunotherapy, and ultimately help to benefit more melanoma patients.